We will continue with our program for today. Next, we have Professor Marc Scheetz, Associate Dean, College of Pharmacy, Downers Grove, Midwestern University in the USA. Dr. Scheetz is an Associate Dean of Research and Professor at Midwestern University College of Pharmacy and holds a joint appointment in the Department of Pharmacology, College of Graduate Studies. Dr. Scheetz is the Director of the Pharmacometric Center of Excellence at Midwestern University. He currently practices clinically as an infectious diseases pharmacist at Northwestern Memorial Hospital in downtown Chicago, Illinois. And we will be listening to his presentation on microsampling as a tool to support precision dosing in children.
I'd like to thank the organizers of CRE Respond for the opportunity to present to you today. Today, I'm going to be talking to you about microsampling as a tool to support precision dosing in children. My name is Marc Scheetz. I'm the Associate Dean and a Professor of Pharmacy and Pharmacology at Midwestern University, and I also direct the Pharmacometric Center of Excellence there. Quickly, for disclosures, I'm a paid consultant of DoseMe, but I don't think I'll be talking about anything today that is relevant to DoseMe, other than samples are needed for many of these modeling programs. Importantly, I have no conflicts with any microsampling device companies. I will be presenting today on behalf of our team for a grant that we have with the NIH. So I'd first really like to start by thanking Dr. Roberts as the organizer here. And back in 2016, their group asked the question, is there a role for microsampling in antibiotic pharmacokinetic studies? And this is a very nice article. I encourage you to read it. And they go through many of the various aspects of how, what, when, where, and why microsampling might be important for pharmacokinetic studies. But if you don't get a chance to read that article, the short answer is yes. It probably does have a role, and I'll show you a little bit today on where I think some of that role exists. So what is microsampling? So if we just want to get down to a very simplistic definition of what microsampling is, we're probably talking about looking at samples on the order of magnitude of about 50 microliters or less of biologic matrices. And really, we're using these biologic matrices for the same purposes that we'd be using them for if we were using large volumes. And the idea there is that we are going to be looking at concentrations of drugs, metabolites, potentially biomarkers, and in a very similar way, but in a much, much smaller volume. And the question has been posed by some groups like AAPS and PBF. They put out a survey and they asked 39 independent organizations, are you using microsampling? And if so, where are you using it? And you can see that from the graph in the lower right corner there, that many groups are using microsampling in a variety of different places. They're using it all the way from drug discovery into their preclinical studies, then into their human studies in phase 1, phase 2, phase 3, and phase 4. So I think the idea here is that they are using these tools in various places. They're using smaller volumes in various places. And the conclusion of this article was that use is widespread and ongoing. But I think they also said that we really need to figure out where there is added value for using smaller samples rather than the traditional large samples that it's greater than 50 microliters of sample. And today we're going to be talking about antibiotics. This was a nice review article put together by Ganesh Murthy when he was still at CHOP and did some work with us. And he showed that when we're looking at dried blood spots, there's actually quite a bit of data on the beta-lactams. So this is a little bit of a busy slide, but ranging from piperacillin and tazobactam to the carbapenems with meropenem, ceftazidime, we're even seeing some data of stability of antibiotics like linezolid, clarithromycin, rifampicin, and vancomycin. And when we start to think about where we can apply this, children seem to be a very interesting place to be thinking about microsampling because they have much smaller volumes. I borrowed this slide from the IRB at Children's Hospital of Los Angeles. And you can see that the allowable amount of blood to be sampled from infants and children up to adult, ranges from 2.5 milliliters per kilogram to 2 milliliters per kilogram within a 24-hour period. When we think about the samples that we take in adults, we're often taking as much as 5 milliliters from a single sample. You can see that this very, very quickly becomes untenable for children. So the answer then is to take smaller samples for some of our smaller patients. And there are many methods to do this. So I'm going to first talk about some of the dried blood spots. One of the most common methodologies in this publication in Analytical Science Advances really, I think, graphically depicts many of these different techniques very well and walks you through them if you're looking for a nice review article. So one of the most commonly used dried blood spot methodologies from the past was using this Whatman filter paper. And the idea here is you put a drop of blood into that circle, and hopefully the blood stays circumscribed within that circle. But as you can see, there's a couple different shades there. Those are different hematocrits. As the hematocrit goes up, it doesn't necessarily distribute as well, meaning that the same volumes could perhaps not take as much space on the paper, leading to potential problems of volume across various samples. There are lots of other methods as well. It was shown in the very center there in B. You can put the blood spot in the very middle, and then it can disperse out to the various places where it could be sampled. And then there are more complex methodologies too, where you put the blood sample in the middle. There's often a spreading layer, and then you tend to have your filter paper at the base. Now, dried blood spots have evolved. There are now companies that are making other polymers that are used for picking up these small samples. One of these is the absorptive VAMS, volumetric microsampling systems. And these are designed to pick up a certain volume of blood sample. We'll show you that in a bit more detail. Now, shown at the right in figure B, you can see that there are also systems where you can place these samples onto something like an arm, push a button, and then that would result in a puncture of the skin, generating small amounts of blood. And then that blood is then trafficked to where the volumetric tips are located for those devices. Now, there are other systems as well. It's not just dried blood that's being used for microsampling. The biggest question, I think, for many of the drugs that we're sampling is what happens with stability. And if we're keeping our samples wet, we have to make sure that we're maintaining cold chain very well and keeping things cold. But there are a variety of methodologies for using our samples in the exact biologic matrix that they come in. So just looking quickly for the volumetric absorptive microsampling system, what we can do is we can draw blood, and then that blood gets placed onto this Q-tip that is the actual device. Putting just a small drop of blood into the syringe, and then the Q-tip is able to absorb that blood up to a defined volume. And then we're able to close that purple plastic lid and then allow the sample to dry, putting it in the fridge and later into the freezer. Just showing you some of these devices that we're using in one of our clinical studies. This is the VAMS format that you're able to pick up 10, 20, 30 microliters of blood. And the precision of volume that we're able to obtain is really pretty good. So we're able to get precision around 5% for these. So the idea here is that you're able to get very specific volumes and repeatable volumes. Devices like these come in multiple different formats that can be either sipped, mailed, or used at the bedside and then sent to the laboratory. And at the end of the day, for all of these devices, it's really important that we have stability for the analytes that we're detecting. In some work with John Williams and Amit Pai and their team at the University of Michigan, they looked at a number of beta-lactams for our study, ceftriaxone, cefepime, piperacillin, tazobactam, meropenem, and ampicillin, as well as vancomycin. And they were able to demonstrate that when they used these VAM systems, stability remained good over up to about 90 days, with meropenem being the sole exception where we start to lose stability and fall outside of the acceptable norms. We talked a little bit about the impact of hematocrit on dried blood spots. That historically has been a problem. It's been something that when you're using the Whatman filter paper has really limited the ability to use those devices. Hematocrit has just been a concern for volumes, what that biomatrix is composed of, and it could have some implications for if drugs distribute to cells. Several authors have looked at this in various formats, and for some of these VAMS devices that we just showed, you can see that for some very common test substances like caffeine, there is a very little effect of hematocrit with various with very little difference in volume with various hematocrits for caffeine. We also took a look in the antibiotics that we're using in our study, and we saw that there was really very good stability in terms of looking at stability of our analyte over time at both the lower end of the quantification curve as well as the upper end of the quantification curve. But we did see that when we were looking in the range between 20 and 60 of hematocrit for vancomycin, we did see a drop. So we did see a drop after precisely between hematocrits of 20% and 30%. We drilled down on that a little bit further, and we wanted to really know for these children, especially the children that are critically ill, do we really lose accuracy somewhere between hematocrits of 20 and 30, because this is very common in our critically ill children. And we saw that really it was only after you got to hematocrits of less than 20 for vancomycin where we started to lose our accuracy based on hematocrit. So the summary of these data, and this most recent one that I'm showing you isn't published yet. It wasn't in the original publication. But we will include that in the publication soon, is that you are able to use these volumetric absorptive microsampling devices in a manner where it's a clinical environment and the types of patients that you would regularly see in children that are critically ill. So the study I've been talking about a little bit is the AMPLE study. This is the study where we're really taking the very traditional PK PD methodologies that I think many of us know so well. We're measuring drug concentrations, and the idea here is to create Bayesian estimates and then later targeted, more precise doses for our individual patients. We've been fortunate in getting a lot of participation from ICUs for children, Children's Hospital of Philadelphia, Nationwide Children's, headed up by Mark Hall, and Children's Hospital of Philadelphia, headed up by my MPI, Kevin Downs. And we've had some good enrollment. Hopefully we'll be publishing some of our data on these models very soon. In very brief, we've been able to show that we are able to model using VAMS for, this is vancomycin data put together by Justin Zhao, who is our postdoc in the lab, as well as looking at cefepime with Victor. And with cefepime, we've been able to show that compared to historic data with plasma estimates, the VAMS estimates for our parameters are very similar. I'll conclude with just a few misadventures or some of these VAMS tips. it is possible to still not take the correct amount of volume for sampling. We have seen an effect called the snow cone effect on a number of our samples that we have not been able to figure out what causes this snow cone effect. It doesn't appear to be hematocrit, but we're starting to work that up in the lab to look at it a little bit further. And then finally, we already talked about the filter paper problems. So in summary, where do we think that BAMs, this microsampling, may be beneficial? We think that really any place that you need to minimize volume and getting accurate volumes would be a good place to employ this type of sample collection. It does and can increase stability of analytes. However, you have to do all of those studies yourself to ensure that that actually is true. It can make the ease of sample collection a little bit easier, potentially even allowing outpatient samples. And then finally, this also allows for a relatively easy transfer to reference laboratories, even from the home environment. So with that, I'll thank you very much and thank all the people that really do so much work on all of our studies. And I'll look forward to your questions later. Thank you.
Well, that was very interesting. We thank Professor Marc Scheetz for sending his presentation.